Hello friends, welcome back to our channel NEET Biology Expert. I am Dr. Parveen. In this lecture series, we are studying Class 12 Biology, Chapter Reproductive Health. In this particular lesson, we are going to see about the last part of our chapter, that is detection of fetal disorders during early pregnancy. So, we have four important techniques through which we could able to detect any fetal abnormalities or genetic disorders or any other problem is there in the developing fetus during the first or second trimesters of pregnancy. So these are the four techniques we are going to study today. So the first one is ultrasound scanning, second one is amniocentesis, third one is chorionic villi sampling, and the fourth one is Fetoscope. So let's see this one by one. So the first technique, ultrasound scanning. What is an ultrasound scanning? This is a prenatal test. So this technique uses high frequency sound waves which are inaudible to the human ear. Okay. So this equipment uses high frequency sound waves. So these sound waves are transmitted through the abdomen via a device called transducer. Transducer is the name of the device which gives this sound waves. Okay. So look at this picture. This doctor is having one object in his hand, right? So this is transducer. So this transducer generates high frequency sound. So when it is kept on the abdomen, okay, so this sound penetrates through the abdomen, and what happens? The sounds are echoed back. Okay, so the sounds are echoed back and they are recorded and transformed into a video or a image form. So this is called as ultrasound scanning. This is the principle of the ultrasound scanning. So very simple. So we are sending a high frequency sound waves through the abdomen using a equipment called transducer. So when it falls on the fetus, the reflection will be there. No? So that sound waves is echoed back. So that echoed sound are transformed into a video or photographic images, and they are recorded in the machine in form of a sonogram. Okay. So this is the basics of the ultrasound scanning. So what are the uses of this ultrasound scanning? This ultrasound scanning we do in different stages of pregnancy, at the first trimester, second trimester, and third trimester. And the need for these stages is also varies. Okay. So for example, in the first trimester, we find out the due date of the baby, to find out the number of the fetuses, okay, and to diagnose if there is any ectopic pregnancy or miscarriage, like this. In the later stages, the purpose varies. So like this. So let us see the overall uses. To look at the uterus and any pelvic anatomical changes like that. For example, if there is fibroids in the uterus, so those things can be observed through an ultrasound scanning. Or to find out the fetal problems, to check the amount of the amniotic fluid surrounding the fetus, to find out the position of the fetus, to check the placenta, its status, like this. There are many purposes for a prenatal ultrasound scanning. This ultrasound scanning is also same like we scan for the other abdominal or kidney and all. No, so almost the same technique. Okay. Right. So what are the advantages of this ultrasound scanning? So here, this ultrasound scanning it is not having any risk because we are not using any radiation here. For example, when you use X-rays, these X-rays are high radiation. They have high radiation, right? So that's why in the X-ray rooms, the pregnant women are not allowed because these X-ray radiations affect the fetus. But whereas this ultrasound scanners, they don't use high radiation waves. They are just using the sound waves, high frequency sound waves. So that's why there is no risk associated with the ultrasound scanning. So only one mild discomfort is the doctor used to push some pressure over the abdomen for the mother. So it may cause a mild discomfort, okay, because the pressure is from the transducer. So it's very normal, right? This ultrasound is having three basic types. So in previous model, that is called 2D ultrasound, which provides just a flat picture of the baby in one aspect, like this. This is the 2D ultrasound scan. Okay, it just gives the flat picture of the baby. And now we have the next advanced method. Because of the technology advancement, we have the next versions, that is 3D ultrasound and now 4D ultrasound. This 3D ultrasound gives the width depth of the images. Okay, so this is highly helpful during the diagnostic technique. So this is the 3D ultrasound, and we have now 4D ultrasound. So that is the next higher version. We can usually see the movement of the baby in the real time in a video format. Okay, so these are the different developments in the ultrasound scanning imaging techniques, right? The next procedure is amniocentesis. So what is amniocentesis? Amniocentesis is a procedure in which a small sample of the amniotic fluid is collected from the fetus. So what is amniotic fluid? It is the fluid that is surrounding the fetus, right? So when the baby is growing, naturally what happens? The baby used to shed some cells from the skin, from the respiratory tract. So those cells will be found in the amniotic fluid. So we are collecting those cells for the diagnostic purpose. So the collection procedure is called as amniocentesis. Okay, so why we are doing this? Generally, we do this to detect the chromosomal abnormalities. If there is any mutation or any problem in the chromosomal abnormalities, those things can be detected using this technique. Okay, right. So this amniocentesis procedure is generally performed in a mother at 15th to 20th weeks of pregnancy. Okay, not early or not later than that. So the apt period for this collection of the amniotic fluid is 15 to 20 weeks of pregnancy. Okay, so let us see how this procedure is done. So first look at this picture. A very thin needle is inserted directly into the amniotic cavity to collect the amniotic fluid. So this is the outer region skin, followed by the fascia, fat tissues, and this pink one is the uterine wall. Below this is the amniotic cavity. So how they are inserting this needle? To insert this needle, the doctor uses an ultrasound scanner. So see here, this is a transducer of the scanner. So first the doctor scans. Okay, so while scanning itself, they do this ultrasound. While scanning itself, they do this amniocentesis procedure. Okay, so the doctor uses the scanning technique and detect the position of the baby first and where exactly the amniotic cavity is present, and in the real time they insert a very small thin needle and exactly insert in the amniotic cavity and collect a little amount of this fluid. So this is the amniocentesis procedure. So here, so let us see what will happen after that. See, first they have to clean the outer area, that is abdominal area of the mother. So clean the abdominal area of the mother or sterilize it. Okay, why to sterilize? Because we know that even for normal injection, the doctor used to wipe the area with alcohol swab, because any microorganisms present on the skin surface may enter into the body, right? So in order to prevent such contamination, so first the doctor will clean the surface of the mother's abdomen and then insert a very fine thin needle and collect the amniotic fluid. Then it is collected in a test tube, transferred to a test tube, and then it is centrifuged. This liquid is centrifuged. What happens when centrifuged? So the cells deposits, because the cells are having a high molecular weight. So the baby cells, these are the cells which gets deposited, and from these cells the DNA will be taken and karyotyping will be done. So karyotyping is what? So to check the chromosome number. So they will arrange the chromosomes from chromosome pair 1 to 23 pair. So like this, 1, 2, 3, 4, like this. They arrange the chromosomes in pairs. So if there is any problem, if there is any mutation, if there is any addition, deletion missing in this chromosome, all such things can be detected in this karyotyping. Chromosomal abnormalities can be detected in this karyotyping. Or similarly, the 23rd pair of the chromosome is the sex chromosome, right? So which determines whether the baby is XX or XY. So the sex of the baby is determined using this amniocentesis procedure. Okay. Let us see what are the uses of this technique. So commonly this technique is used to check the baby's chromosome, that is karyotyping, to detect the chromosomal abnormalities of if there is any mutation. So those things will be diagnosed using this amniocentesis. Okay. The second one is to detect the open neural tube defect, so which leads to spina bifida or open spine. So what is a neural tube? Neural tube is a small area when the baby grows. In the embryology we study. So it develops into brain, spinal cord and other surrounding areas. It will be like a small region like this. Okay. So if there is any defect in the neural tube, it may leads to a condition like this. This is called spina bifida or open spine. So like this it happens. See here. The spinal cord bulges like this. So such babies won't survive for a long period of time even after born. Okay. So those problems can be detected through this technique. Okay. And to examine if there is any infectious diseases are present, or if the family is having any genetic disorders which runs in the families. So if the parents have any doubt whether the baby will also have such genetic disorder, so those things can be diagnosed using this karyotyping, for which amniocentesis is the basic procedure. And also to examine the lungs of the baby, which is ready to breathe, because we know that in the amniocentesis the cells from the amniotic fluid contains both the skin cells of the baby and also the cells from the respiratory tract. So that's how we could see the baby's lung, whether it is developed, how much grown like this. So this is very rare, only we use this. So preferably we do this technique to find out the genetic or chromosomal abnormalities. Okay. Right. So what are the risks involved in this amniocentesis? So unlike ultrasound, which is a very non-risky technique, this amniocentesis is not so. There is a high range of miscarriages associated with this technique. Okay, but though it is said they are very rare, it depends upon the whether the doctor is a specialist or not. If there is any error occur during this procedure or like that, it depends. Okay, so what are the reasons for this miscarriage? If one of the vessels of the baby is cut by the needle, because the doctor is inserting a long thin needle. So while inserting the needle, if any blood vessel of the baby is cut, that time it will lead to miscarriage. But as we say, this is rare because we are not blindly inserting, we are using an ultrasound scanner and exactly looking at the position and then only inserting, right? The second one is may result in an infectious disease. Like as I said previously, if the outer surface of the skin is not cleaned properly, the abdomen region of the mother is not cleaned properly, there is a chance of contamination. Or suppose if the needle is not clean or not sterilized, it may have a chance of contaminating the fetus. So this is the second one. And rupture of the membrane, so that there will be a leakage of the amniotic fluid at the puncture site. So which may leads to spontaneous abortion or spontaneous or preterm labor of the child. So these are the risk factors associated with this amniocentesis. Okay, so sometimes what happen, even the doctor is a specialized person, the baby may also die in some occasions after amniocentesis due to unexplained reason. We don't know what is the reason exactly. Sometimes after this procedure is done, maybe the baby die in the womb. Okay, so these are the risk factors associated with this technique, right? So here we have to understand another important point, that amniocentesis previously was done particularly to detect karyotyping. Okay, so karyotyping, what is karyotyping? In the karyotyping we see the chromosomal abnormalities, right? Chromosomal abnormalities. So in the karyotyping we could see whether the baby is XX or XY. That means the 23rd chromosome says, pair of chromosomes says the gender of the baby. So once people started using this karyotyping technique, so once they found that the fetus is a female child, they used to abort it. Okay, it happened in India. So they used to abort the child. So what happens, at a particular point, the male-female ratio is not balanced because many feticide, female feticide occurs in India. So in order to stop killing the female fetus, which is detected using this amniocentesis technique, okay, in 1994, Government of India has banned this amniocentesis technique. Okay, so this is through an act called Preconception and Prenatal Diagnostic Technique Act. So this act was passed and established in the year 1994. So from that till now, so this detecting the sex of the fetus is a punishable offense. So that's how when you see in all these scanning centers and all, they used to write this in English and also in the local languages, like sex determination of the fetus is a punishable act. Our scanning laboratory will not do this like this. They used to keep a board like this. Okay, so amniocentesis is banned in India. Clear? Now, a third technique is chorionic villus sampling, which is called as in short form CVS. So, what is chorionic villi sampling? So, as how we took the amniotic fluid as a sample to detect the fetal abnormalities, we can also take chorionic villi from the fetus to detect the fetal abnormalities. What is chorionic villi? So we can see here, if this is the placenta of the baby, okay, the mother side of the placenta is there. Similarly, the fetal portion of the placenta is there. So the fetal portion of the placenta is like a finger-like projection, like this. This is what we call as chorionic villi. Okay, this chorionic villi is the fetal portion of the placenta. So what we are doing, we are collecting few tissue samples from this chorionic villi to check the fetal problems. Okay, so this sampling technique is called as CVS. So it has another name also called as chorionic villus biopsy. So biopsy means taking a tissue sample from a live specimen. Living thing is called biopsy. If we are taking the tissue sample from a dead body, that is called as autopsy. Autopsy. Okay, so here we are doing biopsy, right? And another important point, this chorionic villi sampling should be done at the 10th to 12th week of gestational period, not before that, less than that, or not after that. Okay, so for example, amniocentesis we do, let me show you here, this amniocentesis we do exactly at the 15th to 20th week, right here, you remember. So amniocentesis should be done at 15th to 20th week of pregnancy. So here this chorionic villi sampling should be done at 10th to 12th week. Okay, so before amniocentesis, not later that or not early. Okay, so this is the exact time period of gestation. This chorionic villi sampling is done in through two ways. So the first one is called transcervical and second one is transabdominal. Transcervical means this one. The doctor uses an ultrasound scanner and detect the fetal position and see where exactly the placenta is, and then insert a needle like this through the cervix and exactly collect the sample from the chorionic villi from that region. So this technique is called transcervical because the sampling, the root of the needle is through the cervix. The second one is same like as we did in the amniotic fluid, amniocentesis. That means through abdominal route. The doctor cleaned the outer surface of the abdomen and by looking at the ultrasound scanning, insert a thin needle exactly into the chorionic villi and collect the tissue sample from this. So it depends, we can do either way, transcervical or transabdominal. So let us see the uses of this chorionic villi sampling technique. This is used to detect the birth defects, to detect the genetic abnormalities. For example, if the baby will be having cystic fibrosis, if the parents have this genetic abnormality or if it runs in the family, maybe there is a risk for the baby to get cystic fibrosis. So such genetic disorders could be detected using this technique. Chromosomal abnormalities such as Down syndrome can be detected. Any other problem during the pregnancy can be detected. Also here in the chorionic villi samples, we have more stem cells. Those samples are rich in these stem cells. Okay, so this can be stored and further in the future can be used for any treatment purposes like this. This is a very rare thing. People won't do this. Okay, so just to detect the chromosomal abnormalities is the major uses of this chorionic villi sampling. Okay, but this technique also has risk. For example, there is a risk for infection if the needle is contaminated or if the sterilization is not done properly, and if the while inserting the needle, if the baby is disturbed, it may lead to miscarriage or preterm labor. Sometime what will happen if the chorionic villi sampling is done before the 9 weeks. Okay, that's why we say it should be started 10th week. So if it is done before 9 weeks, it may lead to limb defects. That means a defect in the formation of the hands and legs of the baby. Like this problem occurs. Okay, so these are the risk factors associated with this chorionic villi sampling technique. And the fourth technique is the Fetoscope. Fetoscope is nothing but a very simple technique. It is same like as we use a stethoscope. This is not a procedure, just an instrument we use to check the fetal heartbeat. Okay, so we know that stethoscope, doctors use stethoscope to check the heartbeat. So similarly, doctors use a special type of stethoscope to listen the heartbeat of the fetus in the uterus. Okay, so such stethoscope is called as fetal stethoscope, also called as fetoscope in short form. Okay, so this is a device used to obtain information of a fetus within the uterus. So it works in the same way like as a stethoscope, but the only difference is this fetal stethoscope has a bell shaped end like this. This has a bell shaped end like this, like a cone shaped end. Okay, so what happens this, it magnifies the sound waves. It magnifies the sound waves from the heartbeat of the baby. Okay, so it will be audible when we listen it from the outside. So look at the picture here. The doctor is just keeping it on the over the abdomen of the mother with that cone shaped thing. Okay, focusing and just putting the stethoscope on his ears and then listening to the sound of the heartbeat. Okay, right. So previously we have a method. This is called pinned fetoscope. This is just a small cone like a thing. We used to keep it over the abdomen and listen to the sound of the heartbeat of the baby. This is the little older version. Okay, so this is called pinned fetoscope. So what is the use of the fetoscope? Fetoscope is used to monitor the fetal heart rate and other functions during the late pregnancy, and during the time of labor also we use this fetoscope. Okay, so usually the normal fetal heart rate is between 120 to 160 beats per minute. You know that normal adult heartbeat is 72 beats per minute, right? For the child it will be more. For the fetus it will be higher. Okay, so 120 to 160 beats per minute. So if there is any abnormal pattern, high or low in this heartbeat, then it may indicate that the baby is not having enough oxygen supply like that, or any other problem. So to understand this heartbeat, we use this fetoscope. So what are the uses of the fetoscope? It is used to determine or to confirm the fetal position. So in the uterus where the fetus is present, so where the heart is located like that, we could able to determine, and to decrease the prolonged unnecessary ultrasound use in the pregnancy. So before using the ultrasound scanning to detect the fetal position, we can use this as a preliminary technique. Okay, if it is then needed actually, then we can go for ultrasound scanning. So this is one usage. And second, third one is to increase the connection between the mother and a baby. This is just a psychological thing. For example, if mother is coming for a prenatal checkup, okay, the doctor used to keep the stethoscope on her ears and just ask her to listen to the sound of the baby, just to have an intimacy connection between the mother and the child. Okay, so this thing. And during the labor, now we have electronic fetal monitoring. That means not using a stethoscope, we have automatic fetal heartbeat detection methods, machines, that is called electronic fetal monitoring machines. Okay, so that is used during the labor time, so which automatically detects the fetal heartbeat and give its reading in the digits. Okay, so in this lesson we learned 4 important techniques for the detection of the fetal disorders. The first one is ultrasound scanning, second one is amniocentesis, third one is chorionic villi sampling, and fourth one is fetoscope. So I hope this lecture is clear for you. So if you like this lesson, like, share, comment and subscribe to our channel NEET Biology Expert. Thank you.